Welcome to the Theoretical Neuroscience Podcast. I am the host, Gaute Einvoll. The neural action potentials, called spikes, seem to be the key carrier of information in the brain. The spikes have a standardized shape, so the information carried by a train of spikes from a neuron lies in the timing of the spikes. But what is the code? That is, what aspect of the spike trains carry information and is used by the brain to be informed, for example, about the outside world. The simplest candidate code is a spike count code, where the information is thought to reside in the number of spikes a neuron generates over an extended time period, maybe a few hundred milliseconds or longer. This code, often referred to as a spike count or rate code, was, for example, assumed in the work of Hubel and Wiesel in the 1960s when mapping out the receptive fields of neurons in the primary visual cortex. This code thus assumes that there is little information carried by the detailed timing of the spikes. While the spike count code may be what is used in areas of cortex, it cannot be the whole story. For example, precise temporal coding of spikes is required for the barn owl to locate prey based on the sounds they make. To learn about the neural code, I have invited Arvind Kumar. He is a professor in computational neuroscience at the Royal Institute of Technology in Stockholm and has been interested in the question of the neural code throughout his scientific career. In the podcast, we discuss the different kinds of candidate codes and coding and try to sort out the many different uses of the word coding in the field. For example, you have terms like rate coding, temporal coding, independent spike coding, correlation coding, population coding, phase coding, encoding, decoding, efficient coding, predictive coding, and sparse coding. We also discussed some recent results from Arvind's group in Stockholm, obtained in the context of efficient coding, regarding why qualitatively different tuning curves are seen in the brain. A tuning curve is a curve showing how the spike rate varies with the stimulus. For example, how the spike rate in a particular cortical cell varies with the orientation of a light bar. The tuning curves of cells in the primary visual cortex typically have a single peak, but so-called grid cells in the entorhinal cortex have tuning curves with multiple peaks. What can be the reason for this difference? Before we start, I want to do some shameless promotion of our new textbook, Principles of Computational Modeling of Neuroscience, that is just out in the bookstores. The book has a Scottish origin and is written by David Sterrett, Bruce Graham, Andrew Gillis, David Wiltshaw, as well as myself. Actually, this is the second edition of a book that came out in 2011, where I was not involved. But I liked this book and used it in my own computational neuroscience course here in Norway. And when the second edition was planned, I was contacted by the authors asking if I had any suggestions for changes or additions to the first edition. Actually, I had, and after some discussion, I was invited to be a co-author of the second edition, with a particular responsibility for drafting new chapters on modeling of measurements and modeling of electrical dynamics in the extracellular medium, and in brain tissue as a whole. In addition to these new chapters, there are also new chapters on plasticity and model selection and optimization, and the text in the other chapters has also been updated. Of course, I am biased here, but I think this book is a good option to use in computational neuroscience courses or by others who want to learn about the field. As for all episodes, information about the episode and relevant links can be found at the webpage, theoreticalneuroscience.no. And if you want to support the podcast, go to patreon.com/theoretical neuroscience podcast and choose a membership of your liking, for example, at the cost of $1 per month. As a member, you will also get a video version of the podcast recordings, as well as transcripts of each episode. And note that there is a 7-day free trial period when you sign up. I have also made a YouTube channel called Theoretical Neuroscience Podcast, where you can watch the videos as well. I will be most grateful if you sign up to help me fund the production of the podcast. But I also appreciate all the other listeners, and I hope that all of you can help spreading the word about the podcast to other people in the community on social media or otherwise. Okay, here is Arvind. So, welcome to the podcast, Arvind Kumar.
Thank you, Gaute.
So, today, the topic of today's episode is the neural code. So then, of course, you're in the been working on the neural code for many years, and then well, at least sort of many works, many papers, projects related to the question, I guess. So then, I guess the first question is sort of what is meant by the neural code.
Yeah, so different people perhaps read this word differently, but the way I understand is that the whole research enterprise around this question is that which aspect of neural activity is carrying information about external world or
Mm,
internal thinking variables, etc.
mm. But typically, you think about what aspects of spikes, right, or action potentials, or
Right. So I think normally we would think that when we think of a neural code, we are talking about spiking activity of neurons.
uh,
And I kind of, you can call me that I'm a little bit uh, too much into spikes. So I would say spikes are more important, but
mm,
there are people who will talk about neural code in terms of, let's say, local field potentials.
mm, yeah. Exactly.
Um,
Local field potential, which is sort of like this the low frequency part of the
right.
signals you measure inside the cortex. So there's also this related word is which is sort of like neural representations, which is sort of there's a lot of yeah. So what is a neural representation and how does it relate to the neural code?
Right. So again, the way I understand it is that, I mean, if you look at our external world, there are a lot of things that we can read from it.
Mm,
We don't do that. We take whatever is most relevant for the animal to survive, we take in that. And I would say that's the representation. So when you are seeing a visual scene, for instance, depending on it, what context you are and what is your evolutionary history, you will extract different features out of that.
mm,
So I would say that's the neural representation of the external world. And
mm,
now, how exactly those variables are represented in the spiking activity of the neurons, that I would refer to as neural code.
yeah. But like the for example, I think the seminal work of like the Jubel and Wiesel and stuff where they looked at sort of how these different aspects of the visual world and are sort of encoded in this spiking activity of neurons, the receptive fields and so on. So I mean, the receptive field is sort of like a is like a prime example, I guess, of a traditional neuron representation, right?
Um, I mean, in a way, yes, because you would say that, in the retina, for instance, what is being represented is these bright and light spots.
Hmm.
And
Yeah,
as you go further down in the brain, then you see different other features getting represented in the brain, like
yeah.
there are orientations, colors, and things like that.
Represent in terms of their spiking
Right.
activity
Yeah.
and then how
So
neurons
it's
spike.
basically the environment variable which is available in the brain activity that I
Yeah,
would call as a representation.
yeah. So because we don't, I mean, we of course there's a lot of discussion about AI and artificial intelligence, and there we don't. I mean, then you have these deep networks and deep learning, and there we don't talk about the neural code of AI networks, do we? Maybe
Well,
we should.
actually, we don't, and the reason is that we exactly know what it is.
Okay. Yeah.
We do talk about representation, though.
Yeah. Yeah.
So, in terms of, like, let's say that you have a deep net which has been trained to classify images.
Mm,
There, we exactly know how pixels of an image are translated into the inputs to different neurons in the first layer.
mm,
So, there's
yeah.
no need to talk about
A code,
a neural code.
but it's
We
also
talk
a thing
about
that it's
representation. We talk
yeah.
about representation in the
Yeah.
sense that we ask these questions, that which aspect of the image
Huh.
is
Mm,
available in one of the hidden layers, or hidden layers of the new deep net, which aspect of the environment they are encoding or responding to.
mm,
So we do talk about these terms in terms of
yeah.
representation. So basically, the part we don't know, we discuss. The part we
Yeah.
know, we don't.
But I also feel a little bit that when you're like in the retina where you have these graded potentials, you don't have spikes, and also in the artificial networks, you also have like one number. You don't have spikes. You have like in some sense like a graded potential. But code, then you will, I think sort of like Morse code, right? Something which is sort of discrete and like spikes. And sort of
Yeah.
that we think about codes, we are more used to this binary codes and yeah, something where you have like this very standardized spiky representations.
Yeah, I have also a feeling that, when we use the word neural code, somehow we create another connotation. And that's what I
Mm,
initially meant, that some people mean different things with neural code.
mhm, mm,
And there exactly this confusion arises, that if it's a code, like it's a
mm,
Morse code, or ASCII code, or some other fancy code, so
mm,
you kind of believe that there's a decoder which who knows the code. You have an understanding how to
mm.
decode. It becomes more like cryptography than.
Exactly. You have to crack the code.
Yeah, exactly.
Exactly
But
like
I
this.
don't think that's the right connotation, at least in neuroscience, when we say
Mm,
neural code. At least my understanding is that since the days of Adrian and so on, when we started to talk about what aspect and how neurons are encoding the environment, we were more interested in which features of the neural activity is
mm,
used for that.
mm. So, I mean, you wrote the paper together with, I guess, Stefan Rotter and Ad Edson, yours and my friends down in,
Yes.
in Freiburg. I guess it was in 2010, which sort of focused on this, well, on coding, at least some aspects of this coding. And there you refer to this paper by, I think it was from 68, by Perkel and Bullock, where it said something. You had this little, it was like a review paper. You had this little box about the four functions that a candidate neural code must serve. Do you remember this? This
Yeah,
like
yeah, I do remember, and I think
the school
it's a
exam.
it's a very useful guiding, or let's say these are the nice boundary conditions for a
Mhm.
neural code, actually.
Okay.
But just to make this point, that this is not really a paper from Perkel and Bullock. It's actually um proceedings, pretty much. It's a summary of a big meeting they organized in '68 about neural
Hmm.
code.
Hmm.
So there are
I
not just
see.
they talk about boundary
Mm,
conditions of codes. They also refer to various features of
mm.
or various possible codes that people were discussing at that time. And in some sense, it sounds very embarrassing now, but we really haven't come out of that yet. So we are still following basically the same ideas, checking their viability, feasibility, etc.
Yeah. By the way,
Uh,
so yeah, I just want to say that this review paper that you wrote, this was in Nature Reviews Neuroscience 2010, will be sort of I'll be well. I mean, if you look at the web page for this episode, you'll find a link to it, so then people can also find this reference to this Perkle and Bullock paper or this conference report or whatever.
yeah, I strongly recommend this. I think every student of neuroscience should read that.
Cool. But then back to the
Yeah.
four
So
functions.
these boundary conditions, they argued. I mean, they make very much sense when you read them. That it should be possible. So whatever variable you pick up from neural activity or brain activity, you want that it can encode. Meaning, stimulus should be able to change it. So
Hmm.
that's the representation in that. Like if
Hmm.
you choose spike rates, for instance, if it turns out that spike rates can't be mutated because the stimulus conditions change, then you know that this is not a viable
It can't
code.
be muted. It can't be changed. You mean?
Can be
Yeah.
changed,
So
yeah.
yeah. So if it can't be changed, yeah,
Yeah.
exactly. Then it's not
And
a
then
good.
it should be possible to interpret it. For instance, whatever activity exists in the brain, if it is not translating it into something visible, let's say motor output or some other response, meaning one brain area interpreting another, if you can't interpret it, then also it's not a good idea.
Mhm.
Um, and
And
then they
because
talk about
especially the upstream or what do you call it,
downstream.
downstream neurons, the ones
Right,
that
like
receive
V2
input.
should be able to interpret what V1 has
Yeah.
done.
Mm,
Muscle should be able to interpret the motor commands and so
exactly.
on.
So it must be interpretable.
Right,
Exactly.
interpretable. And then we do want processing. We just don't want a kind of sensory motor representations.
Mm.
We want to process it. We want to modify it depending on the context, depending on what we want to extract from it. So
Yeah.
that's the idea of transformation, that we should be able to change it, sort of the processing metaphor, if you wish.
Yeah.
And
Exactly. Without it, you wouldn't really be able to do computations
exactly.
and make decisions and
I
stuff. You
mean,
mean?
the least we want is that, for instance, we should be able to merge different type of inputs to create a composite,
Yeah.
right? An
Mm.
audio visual, for instance, or
Yeah. Mm.
and then they talk about something that often people miss. That was the transmission aspect of it.
Mm.
So, in some sense, it's in the interpretation, but they wanted to make a case about it, that neural activity has to go from one brain area to another brain area, one region to another region, and
Mm.
eventually come out as the muscles at the muscles. So it should be possible to transmit it.
mm,
Now this is nice because, like for instance, we could say that, let's say, membrane potential of a single neuron, that's actually, it's perfect. You can modify its variance and mean and other properties. You can interpret it, you can transform it, but you can't transmit it.
mm.
Membrane potential of a neuron is
Uh,
private to that neuron,
yeah.
and only thing that goes out is spikes. So in
Mm,
some sense, these boundary condition, the transmission aspect automatically excludes membrane potential as a potential code. Then.
mm, mm. Yeah, no, that's a good. That's why God invented spikes or
Yeah, well,
axons, so that they can sort of, yeah,
yeah.
so that they could transport it. Exactly. So okay, so that's and then sort of like, and then if you go into like different code candidates for the code, and I guess the oldest one, or the ones that was first seen in experiments, was just also called the spike count code. So what is a spike count code?
So what Adrian observed was that I
Yeah,
think the
so
muscle activation was proportional
yeah,
to
so
number
this
of.
was back. He was really a pioneer. It was back in the 30s or something, 20s,
Uh,
30s.
yeah, I think this finding was in, if not late 20s,
Yeah.
before the war for sure. I think.
So then we had finally like we had a good enough amplifiers or whatever
Yeah, I think
equipment, so
so.
you can actually could see these spikes.
Yeah, but he saw it only in the periphery. I
Yeah,
don't exactly remember what was the experiment
periphery
now.
out in the muscles or something.
Yeah.
Yeah,
So
there's something with
basically,
a
you
frog, wasn't it? No, or maybe.
frog,
Yeah.
very likely frog.
Mhm.
Um, and the amount of current you inject, the extension of the muscle was somehow proportional to that, and the
Mhm.
number of spikes that you were seeing in the motor neurons, I
Yeah.
think something like that
So the
was.
more you extended pull the muscle or something,
Yeah,
then you
more the
get more
spikes,
spikes.
more
Yeah.
the extension, something like that was there. So
Yeah.
that
So what is
starts
a
this notion of rate coding or spike count
yeah? So
code.
that's exactly. Well, let's stay with spike. Well, I want to talk about rate
Yeah,
later. So spike count code. That's the
yeah,
number of spikes
number of spikes,
within a time interval,
exactly.
and maybe like a few hundred milliseconds or
Yeah.
something. Well, which is often
Mhm.
used. And I guess that's also what was used in. I mean, for example, in Jubel Wiesel, they also looked at spike count codes mainly, didn't they? They sort of, for example, these simple cells, orientation selected cells, they counted the number of spikes within a
Yeah, I
certain
think
time
ever
window.
since the days of Adrian, this is what we have been doing. Like, this is the
Mm,
first analysis that we perform
mm, yeah,
already from ganglion cells, retinal ganglion cells,
yeah,
LG and V1.
yeah.
Everywhere we count spikes in response to
Mm.
some stimulus.
And this is where AI is at the moment, right? They also produce one number as an output.
Yeah, it's more continuous though.
Yeah, yeah, that's true. Maybe. Well, I guess it. Yeah, but at least yeah. So okay, so then if you then so then you have for like a single neuron can sort of we have the
Yeah.
spike count code, but then you can also then start to look at the time resolved version. You can sort of count the number of spikes within within certain time windows, right?
Yeah, I mean, you mentioned earlier technology, right? And I don't know if I'm just straying too far from the podcast, but
Oh,
it really
we like to
reflects
stray a little bit.
it really reflects the kind of technology we have, right? So very
Mm,
early on, we don't really have this opportunity, to record spike trains and analyze them and so on. We just get, okay, on the oscilloscope, you see so many spikes.
mm,
Maybe you can measure the interval a little bit, but not too much. You're recording a single neuron. So yeah, spike code.
mm,
And as the technology evolves, we can record over time and analyze it further. So all other codes evolve basically as the technology evolves. Correlations came up when we could record two neurons simultaneously.
mm.
And nowadays we don't really talk about it because we can record thousands of them
Mm.
simultaneously.
But
So
even in one, even if you just focus now for now on a single neuron,
yeah,
so then you can also then record many. I mean, you can sort of follow the time
yeah,
delay
yeah.
of when this neuron spikes, and then you can from that, if you have a time resolved like measurement of the spikes, then you can sort of convert it into a firing rate, right?
Yes.
And like this, like yeah, how do you do that?
So the simplest idea is that you take the inverse of the interspike intervals.
Mm-hmm.
Otherwise, you
So
bin your
inter-spike
time
interval,
into.
that's just the intervals between two
Yeah.
spikes. Mm.
Mhm.
Hmm.
Otherwise, I think most common approach is that you divide your time into certain fixed bins,
Mm.
and then you say, okay, how many spikes I see per bin, and that becomes the spike rate then. So
That's almost
instead of
like a
like,
little sort of
if
like,
you look at the experiment over 1 second, for instance, instead of counting all the spikes, you say, I count it over windows of 50 milliseconds,
mm,
and that becomes my rate.
mm,
It kind of becomes a bit arbitrary then,
yeah,
what resolution you are choosing.
yeah,
And that becomes a very tricky issue in terms of definition of the rate code itself, right?
yeah,
What is a spike rate?
exactly. And yeah, and it's so then maybe it's you can start thinking of it as sort of like a probability for spike firing
Yeah. Mhm.
instead of like an actual measured
Right,
right,
but
yeah.
there also, I think we are forced to stick to spike count somehow. We
Mm,
have to take into, like, we can't. I mean, we will become then
okay,
frequency. We'll just
frequentists,
look at the frequency and then convert
yeah,
it into probabilities.
yeah. So, but then of course this is if you have a single spike
Yeah.
train, single measurement. But if you then you often do this, like especially if you work in the visual system, or I guess if you work out in the periphery or in the sensory system, you often do this trial averaging, that you do the same experiment many times, and then and do some kind of average to get this what they call
Mhm.
peristimulus time histograms, or you sort of pull the spikes from different trials.
Yeah, but before that, actually, even for single neuron, we can talk about the latency code.
Okay.
So not just the intervals, but like time for the first spike. So
Yeah.
I mean, we magically assume that somehow the brain knows when the stimulus arrives.
Mm,
But typically in the periphery, you can imagine that there is a very fairly low activity and
mm,
pretty much silent. So in many examples, sensory transduction, particularly like even touch, for instance, turns out a lot of information is carried in the time for the first spike. When the first spike occurs in a neuron,
mm,
with respect to some arbitrary reference point, of course. As an
mm,
experimenter, we know that reference point, and therefore we can comment about it.
mm, mm.
But
Oh,
there is information in that.
yeah.
Even in retina, it turns out that in salamander retina, you can read a lot about the stimulus contrast just by looking at the time to first spike.
Mm,
But maybe the brain could create these reference internally.
mm,
So one could argue that even that is useful way to encode information.
mm. Yeah. And then you have the trial averaged
Trial
way. Yeah.
averages.
So then, but that is sort of like that's more like a measurement
It's more a
trick,
measurement.
right? Measurement trick,
Initially
right?
started as a
Because
measurement,
yeah.
given that there is so much noise in the
Mm.
system. But then people
Noise
argued
meaning,
that,
what do you mean by noise here?
meaning that at least in the cortex, it turns out that neurons are not silent
Mm.
when there's nothing present. And well, I mean, it's an active brain,
Yeah.
so neurons are doing something. We don't know what it is.
Yeah. So it spikes even, yeah. So it spikes even if it's no stimulus that you have put on or
Exactly,
stimulated with. Yeah.
exactly. So that's why I'd introduced this term called ongoing activity. That
Yeah.
instead of calling it noise, there is an
Mm.
ongoing activity.
Addis and odd Addison, right?
Yeah. Mhm. So they coined this idea of ongoing activity, but then if ongoing activity is different every trial,
Mm.
you can't really be sure what's going on, so you average it out.
Yeah.
And then they found out, this was
You
also
sweep
funny,
it
that,
under the carpet.
yeah,
I
you try
think,
to,
which you also said, right? I think Adi used when we
yeah,
had
yeah,
the,
this is what they did in the beginning. And then
yeah,
when they looked under the carpet, they found out that actually this variability is also informative.
yeah, yeah.
So
So exactly. Mm. So that's the trial average, and then you get this what's it called? This whatever. I think it's peristimulus time,
PSDH,
PSDA.
PSDH.
Exactly. So that's in some sense, then you can get this time resolved firing rate,
Yeah,
right?
but I just wanted to add there that some people then argued that, oh, you as an experimenter, you can do multiple trial, but a brain cannot. So
Absolutely.
the counter argument to that was that the brain has many neurons, and
Mm.
you average over many neurons in the single trial.
Exactly. So this is sort of then you're over to these populations,
Right.
where you also then you have also this. I mean, for example, like this spike. Well, for one thing, you can have a spike count code, obviously for populations,
Yeah.
and I guess that has been used. Hasn't that been used sort of to, especially then to decode motor in the motor
Yeah.
system?
I mean, starting with the work of Hubel and Wiesel, so obviously they found what they call then the tuning curves, that neurons they respond maximally to some
Yeah.
small range of
So
stimulus.
just define it, because the tuning curve is an important concept. So what
Mhm.
is a tuning curve in the concept, for example, in the context of Hubel and Wiesel?
So in the experiments of Hubel and Wiesel, what they noticed was that if they present a moving stimulus, neurons respond. And with that, they could map out the receptive field and in which region of the space the thing should be moving when they respond. But within that region of receptive field, unlike, say, retina, now it matters which orientation this bar takes. So neurons, even though it's their receptive field, they don't respond to all ranges of the stimulus values. They respond to a narrow range. So like a neuron is responding maximally for 90 degrees and a little bit for 75 and 125.
Exactly.
And then it
So then,
tapers off. And since it was going like non-monotonically, it was peaking for some value and going
so
down
you're plotting the response as a function of some aspect of the
some
stimulus.
stimulus value.
Yeah.
Yeah.
So okay, so that's the tuning curve. So and then okay, now back to Jubel and Wiesel. I think I interrupted you when you talked about population code.
Yeah, so they had these tuning curves, and if you look at one tuning curve, it's as you said that it's a response plotted against the stimulus value, but it's non-monotonic. So that means for same value, you can have multiple. No, same firing rate or spike count of neurons could mean multiple stimulus conditions. So how do you disambiguate that? And then the idea is that well, neurons have different tunings, and you take the whole population as an ensemble. Some neurons are tuned to maximally, let's say, 90 degrees, others are for 75, 45, and so on. And this gives you an array of values. And from there, you can create a linear decoder and estimate the stimulus in an unambiguous manner.
Exactly. So this they used, and so that was the idea from Jubel and Wiesel, and this was used. It wasn't in the
So I'm
motor
not sure.
system to sort of decode the arms of monkeys or something.
So I'm not actually sure whether Hubel and Wiesel talked about this kind of decoding. They were happy with explaining that this is how the brain encodes.
Yeah.
I think it was, as far as I know, I think it was Georgopoulos who first showed it in the motor system that you can decode the movement direction from this.
Yeah, because I guess it's exactly. And so that was
And then it
measurements
became
on
population vector. So you create a vector which points in some direction depending on the tuning of individual neurons.
And where each axis in this vector space, whatever, it corresponds to the activity of one neuron.
So each neuron makes one dimension of it, and then you look at the activity as a vector in this, in which direction it points. That should be the direction of the.
So, I mean, and of course, then you can also take this further on to get some time-resolved firing rates, time-resolved population firing rates.
So then you can do it in a more or less continuous time, looking into small time windows.
And there it's easier. You don't have to necessarily do this trial averaging, right? Because, well, at least if you have many neurons that you average over,
you are averaging over many neurons.
Yeah, so you get some kind of signal at all, like you get sort of like a time resolved signal,
right?
Automatically in a way.
And that created another issue, right? So when people started to do that, they realized that there is a problem with this idea because somehow population code or population vector requires, or it works the best when the individual neurons are unrelated to each other, uncorrelated. And it turned out that there is a fair bit of correlation in the neural activity.
Yeah, we're coming to that. We're coming to correlation a little bit later, because I thought maybe you should start with if you sort of look at if you do this, have some estimate of back to the single neuron firing rate, and then you have like one possibility, sort of like to interpret it as a probability of firing an action potential at different points in time. And then there's one possibility, right? And that sort of these individual spikes encode information independent of other spikes from the same neuron, right? So it's like, and that is called an independent spike code. So and then of course it's and then you have the other. You can also have that it's not only independent of the spikes of the same neuron, but also independent of the spikes of other neurons. So meaning that I guess that means that there's no correlations. And that correlations has to do with the relationship between spiking patterns of different neurons, right? So I guess that independent spike, both between your own, this independent spike, if it is an independent spike situation, both in terms of relationship to your own spikes spikes in your own in the same neuron and other neurons. That is, it's really, it could be called an independent spike code, and that is maybe, is that, could we call this a rate code from here on?
No, not really. I think it would still be called a spike code.
Okay.
Um, and I actually doubt how feasible this is, this kind
Okay.
of individual spikes, because whatever we know about, so maybe in the sensory areas, early sensory periphery, this may be
Mm,
true,
mm,
that individual, let's say, occurrence of a spike at a given time is
mm,
indicator of something.
mm. Yeah.
But remember, it's a zero or one event, so it
Mm,
can only encode then presence or absence of something.
mm.
And if it needs to do something else, then it needs already one more spike to take reference to.
Sure. No, I was more thinking about like the naming thing. I mean, like whether it's used or not in the in real system, but could we, just, it's more about sort of keeping the terms
Oh, yeah.
separate. So is it okay to call a independent spy code for a rate code for now, for the rest of the podcast, or okay,
I
maybe
don't know. I think
why don't why would, because at least alternative, if there's information in the spike train beyond the, like the like beyond the just the probability for firing, which is in
yeah.
this independent spike code, then that is maybe called correlation code. I'm sorry, I read this I think this is in a very nice book by Theoretical Neuroscience by Diane and Abbott. They have this, I think, in the first chapter, where they, I think they go through like have a nice description of how this, they use this called independent spike code, and contrast it with a correlation code, that is information beyond what's in their independent spikes
Yeah, so I
separately.
would actually stick to that terminology. I wouldn't try
Okay.
to put an extra label on it of
Okay, we
rate code. I
stick.
would,
Okay, so then we stay with independent spike
yeah,
code and correlation code.
correlation code.
Yeah, very good. Okay, so then, and then if you sort of, but then if I look at sort of your this review paper that you wrote together with, Stefan and Ab, then you study this, I guess, transmission of spikes in these recurrently connected networks, maybe like mimicking the cortical networks, where you have sort of like a bunch of spikes propagating from one area to another. So I guess you focus particularly on this transmission aspect, the fourth point in this
Yeah.
list by Perkel and Bullock. So, but there you used different words. Then you used asynchronous code and synchronous code, right? And I guess also you call the asynchronous code, you sort of say, well, that's sort of like a rate code, and the synchronous code was a temporal code.
Mhm.
So I don't. How does this relate to this? Well,
Right.
I guess we can start maybe with synchrony. What is synchrony?
I think this goes back to this idea that you started with, like independent spike code, for
Okay.
instance. So there you say that a spike is independent of any other spike.
Mm.
It's only, let's say, driven by the input, which has some value.
Mm. Okay.
Now, it turns out that spikes could be dependent on each other. It may be difficult for a neuron
So this
to,
first thing, I mean, just like, so if it only depends on the input, some kind of stimulus input, but
yeah,
it doesn't really, it only feels the input, not the other spikes, or not the other neurons, that you would call, that's like asynchronous
yeah,
or
that's completely independent, right? And
independent. So that's I guess that's what, yeah, exactly. So that's an independent.
so I want to use a slightly different terminology here. So if I'm in a single neuron,
Mm.
and then I see that spikes are independent, then of course it's independent spike code. However, if they start to depend on previous spikes,
Mm.
then sometimes we use the word serial correlations or
Okay.
temporal correlation.
Yeah.
That there's a time correlation or serials, this
That's like autocorrelations, maybe.
autocorrelation pretty
Yeah,
much.
exactly,
Yeah.
because it's with
Yeah,
yourself.
exactly.
Mm.
So this
Okay.
autocorrelation is, if it is there, then there is some other information, right?
Mm.
Maybe you are merging the previous inputs with the
Mm.
current input and so on.
But this goes beyond the, say, if you have this firing rate input, or you have this input. So at some point you have this firing rate which is higher because you got like a stimulus that you liked, and at a later point it's lower. So then you also get this, you get autocorrelations just from the fact that you have this underlying firing rate, but it's not really, they're still independent in a way, right?
Yeah,
Yeah.
absolutely.
So that would be an independent
Right. So
spike code.
it should be independent of the stimulus, let's say spectrum.
Okay. What do you mean by that? No.
Meaning, if the stimulus is changing fast enough, then of course your rate will change,
Yeah.
and your autocorrelation function will reflect the change in the
Yeah.
stimulus.
Yeah.
But
Exactly.
what you want is actually something faster than that, I would
Okay.
say.
So you want something beyond that sort
Yeah,
of
right.
just the firing rates are changing.
Right. So that's one kind of correlations
Yeah.
that can appear.
Hmm.
The other kind of correlation that appear is that because neurons in the brain are connected, they share their inputs and so on. So therefore, their spikes are not independent.
Hmm.
So that becomes another kind of correlation. And if
Yeah.
you wish, that's the spatial correlation,
Hmm.
because neurons are arranged in
Well, that's
space.
what we call a cross correlation.
Right. That's what you would call as cross correlation.
Hmm.
And this is something that is then referred to as synchronization, that
Yeah.
neurons are synchronized to each other.
Yeah. If the kind of correlations well,
Right.
I guess if they end up firing,
Even if it's small correlation, right? That's sufficient to say that something is encoded in the correlation.
yeah, exactly. So that goes beyond then, because you could have two neighboring neurons that just gets independent, well, gets so like some kind of stimulus input, which makes that the firing rates of these two neurons
Yeah,
go up at the same
absolutely.
time. But that would you call that a correlation?
No, that's not really correlation. So there is
No.
another terminology that
Yeah.
comes in there.
So exactly. So the cross correlation is something that goes beyond
Yeah,
that. They have so like co-varying input in some sense.
exactly. Right. So this is something that we corrected in the autocorrelation as well, that it should be different from the stimulus
Mm.
autocorrelation.
Yeah.
And in here also, we want to exclude that,
Mm.
that it should not be just the signal induced correlation.
Okay. That's quite complicated, but you
Yeah,
have.
yeah, so this layer after layer, the new
Yeah.
terminology keeps adding, like we talk about signal correlations and
Mm.
noise correlation.
Mm. But I think in sort of your in when in back to this paper again, when you talked about like some asynchronous code, this was some kind of, that was something that was contrasted with the synchronous code, and so that had to do with the temporal resolution of it. As I mean, you also, I think you call the synchronous code for a temporal code.
Yeah.
So
So
that is something which is sort
it
of
becomes
varying at
muddy.
the.
Yes, I
Yeah.
agree. So the idea was, since we start with this notion that spikes are independent,
Mm.
so then you can use the terminology of asynchronous, meaning individual spikes are asynchronous with respect to each other within the single neuron and across neurons.
Mm.
And this is then also becomes a more clean definition for rate code, that when we talk about rate or count, if you wish, we
Mm.
only talk about count. There's no extra information to be gained by arranging spikes in a specific temporal order.
Mm. No. Well, but I mean, you
And
could have like at the count, but you could also have like a slowly varying
yeah, but then that
firing
will
rate.
be imposed because of the stimulus conditions
Exactly.
changing,
Mm,
right? Like if
exactly.
the contrast is changing. Yeah,
Exactly.
of course.
Mm. So it
Uh,
follows the stimulus, but it's not like nothing beyond that sort
yeah,
of.
nothing beyond that.
So that's something you call an async. So then there's no
Then
correlations.
then we say, okay, it's asynchronous, but that's part of the definition of the rate code itself. That
Mm,
underlying assumption is that the spikes are uncorrelated,
mm, yeah, and it only follows the stimulus input,
only follows the input.
and
Yeah,
that's a rate code.
that's a rate code when you can jitter the spikes
Yeah,
by small amount.
but isn't that like an independent spike code then?
Well, it is, yeah, exactly. It's in the word independent, right?
Yeah.
That the
Mm,
independent meaning it is asynchronous with respect to everything else.
yeah. Okay.
So I now I can't remember. There was once a thought experiment suggested when people were talking about, whether the timing of spikes matter or not.
Mm,
They said that what if there's a drug
mm,
which can jitter every spike by small amount,
mm.
and if the person falls down, then we would say that the spike timing mattered and not.
I see. That was a cool thought experiment.
I think
Uh,
it was
maybe I have
Scats
two.
or somebody,
Yeah.
soft key maybe. I think
I'm just
somebody
thinking of
like that.
like this. I mean, of course, we have backgrounds in physics, so we remember all these thought experiments that was at the 100 years ago when they tried to figure out sort of quantum physics and understand that. So they had
Yeah,
all these cool thought experiments, in particular Einstein, I guess, or maybe it's
yeah.
just been
I
this
think
sort
it's a
of
very nice Gedanken to argue
yeah.
about.
So maybe we need more thought experiments in
Yeah,
neuroscience.
I
Okay.
agree.
Yeah. Okay, good. So,
So
mhm,
so yeah, that's what we refer to as that. We have an ensemble of neurons. So okay, this also I want to clarify that since I grew up in Freiburg for my academic training,
mhm.
um,
So, what
we
was it?
went.
You studied first in India,
Yeah, I
and then
did
you
my.
came to Freiburg in South Germany,
Yeah.
Southwest Germany. Mm.
So I think very quickly it was realized that individual neurons are maybe not so useful because
Ah,
the connections are not very strong.
mm,
Moreover, there is this convergent divergent connectivity, so individual effects may not be visible. Therefore, you need to talk about many neurons simultaneously, which was already reflected in the population vector idea.
mm. Yeah.
Right. So when
Mm,
you talk about an ensemble of neurons, so this is how I would like to think about neural code. Then that you give me a bunch of neurons with bunch of spikes.
mm,
How do I arrange these spikes so that I can encode some information in them? I could do it by increasing the rate. I could do by some temporal patterning and so on. So one extreme we took was rate code, where spike timing doesn't matter. I can throw in those fixed number of spikes
mm.
anywhere in this
Yeah.
box, and that's it. It doesn't matter. It's like gas molecules. They could be anywhere in the room randomly. The
Ah.
temperature is unchanged.
So it's just the total number of spikes at each time that matters.
Yeah, exactly.
Mm,
I think
population
that's.
average. Yeah.
But then you could say that, okay, no, 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 there's maybe more information in the way I arrange these spikes together. So one extreme of that was um, what At and Moshe used to call as pulse packet, that you
Mm.
take all the neurons and you
Moshe
say
is Moshe Ables,
Moshe
right?
Abilis. Yeah.
Mm, mm,
So what they introduced was this notion of pulse packet, which they said that, I mean, just as an extreme, that all the neurons, they spike within some temporal window together.
mm,
And that's an information. It's
mm,
not fast rate. It's more than that.
mm,
And that was then the what Moshe used to call as the Sinfire,
mm.
uh, because he needed to introduce this idea because of this whole notion of weak connections and convergent divergent
Yeah.
projections.
Because the Sinfar ID is sort of interesting. That was like this ID, I guess, which was originated with Moshe Ablesen, that there was like this he could sort of there was like this very precise time, something
Mhm.
about the times that that was really reproducible, some spike timing or something that was really reproducible,
Yeah,
or that could sort of go over long times. And I guess this it was interesting sort of how this could maybe be implemented in real networks, even though you are in
exactly.
this regime where in the cortical where you have each input is quite small and you
Yeah,
need
just
many inputs. And
exactly what they noticed was that in some examples, two neurons, they were spiking with
mm,
like sub millisecond or like millisecond precision,
mm,
even though the distance between their spiking was like tenth of milliseconds or
mm,
hundreds in some cases.
yeah,
And
mm,
Moshe argued that that can only be possible when neurons spike together.
mm, mm.
Otherwise, individuals are very weak.
So that's sort of like, so then you need this pulse of spikes.
Yeah.
Mm,
So then these are the two extreme cases. In one
mm,
case, there's no information in the spike time.
mm,
Here, you require that all of them spike together.
mm.
So in
Yeah.
some sense, this pulse packet idea is just a amplified spike. It's 0 or 1.
Mm,
It
mm.
doesn't have
I
more
see.
information than
Yeah.
that.
Because
But
I guess, yeah.
these are the two extreme
Yeah.
cases. And other codes, and I believe that the realistic or the final answer perhaps to this neural code problem is somewhere in between these two extremes.
Mm,
Um, so that's why in that time when we were like, I think it was 12 years ago or so, we were thinking about like what are the limits on these two extreme form of codes.
mm,
What can I do with this rate code? What can I do with this pulse packet like code? And it turns out that, and what people had missed was this mostly the transmission aspect. So when we put it together, it turns out that the rate codes are actually not easy to transmit.
mm,
And the reason was that if you say that one group of neurons, they are firing asynchronously, but these group of neurons will project to another and they will crisscross in their connections. And because of that sharing, this rate signal will become correlated.
yeah.
So either you need an active mechanism to decorrelate the activity,
Mm,
or you have to say that, well, this is not a feasible form of code because it loses information as it goes one stream
mm,
up
yeah.
or down.
Mm,
So this is what we try to argue there, that there are these two different codes and they have problems. But it turned out that when you think of transmission, there is a small region of parameters where you could say that, well, both could coexist.
Hmm.
And that was a nice answer in
So
the
the
sense that,
parameters there are like synaptic weights and how
yeah,
much, like that's what's called convergent and divergent
yeah.
connections,
So,
and
I
sort of how
mean,
much to.
so yeah, I mean, we were really thinking about the neural hardware at that time. Like, okay, I have this kind of neurons, such as the connectivity, given these constraints, under what conditions a particular code will go through or not.
Mm,
And since we worked with two extreme forms, we could say that, okay, this is what is feasible and not.
mm, mm,
But since then, this has been, I think, one of the ways I try to think of the problem of neural code is that think of it in the context of the brain's hardware. Most of the time, when we think of neural codes, people
mm.
talk about transformation or encoding, decoding. They're only worried about how much information you can extract from them.
Exactly. So this goes a little bit back to this sort of like this very sort of like intuitive and maybe well incorrect notion that there's some kind of homunculus, some
Yeah,
kind of infinite genius sitting in there
yeah,
and it's extracting information and all the information that is in principle
exactly.
in the spike trains. But that's, as you
Yeah,
said here, that's not the case, right? You need some kind of code that can actually be read out
I mean,
downstream.
in the spirit of Maxwell, I could call it there's a spike demon.
Okay, exactly. Yeah,
Uh,
exactly.
But
Mm,
this is, I think, also a problem that Roman Brett mentioned in his critique of neural code.
mm,
So he wrote a very nice article called "Is Neural Code a Good Metaphor for the Brain?" or something.
mm,
And
mm,
there, this is the problem he's asking that somebody must know when the input starts, when the input ends, what's the code, how to interpret it.
mm.
Yeah, I think that's maybe taking this idea of encoding, decoding too literally
Yeah.
to say that, hey, we are assuming an ideal observer, and that ideal observer can do magic.
Mm.
So
So
my
yeah.
take has always been that eventually this decoding must be done by neurons, by downstream
Mm,
networks.
mm.
And somehow this connectivity between the two networks and the local connectivity within the receiving network must play a role.
So now you're into this. I mean, that's another sort of. I mean, we talk about encoding and decoding. So that's another word use of the word coding. So what is encoding and what is decoding?
So encoding is that, let's say that I have some stimulus variable,
Mm,
let's say angle 90 degrees, and I want that neurons change their spike behavior in some way, so
mm,
that at some point I can extract 90 degrees out of that spiking activity. So encoding would mean that changing the neuron spiking patterns
mm.
uniquely, hopefully, that you can recover it.
Yeah. So that's sort of like and like this is sort of like the forward modeling, right? That's typically when you do then you model encoding. If you want to make a model for a neuron or a network, then you typically end up computing and calculating, predicting spikes or action potentials, and then sort of like so that is a model for the encoding.
Yeah, so you could actually use this terminology as well, that encoding is a forward model
Mm.
and decoding is an inverse model,
Yeah, but because decoding, then you take the signals
and then try to
and then
recover.
yeah, yeah. So and that's sort of like yeah, and then the point is that sort of often when you look at the people that have worked on decoding, then they look at this optimal decoding techniques and say and if you're sort of like this this spike demon in there
Yeah,
who's like infinitely smart, what could he or she get what could a demon get out of the spike train? But that's
yeah.
not necessarily possible for the.
I mean, in a way, we can sympathize with that view because, I mean, we believe that, or there are proofs that neural networks are extremely powerful, right? They can do
Mm,
amazing
yeah.
computations. So, in principle, you could imagine something like that. However, that's really far fetched. I mean, you require very
We
specific
also need
connect.
to, I mean, in this, when we, all of us in this computation neuroscience field, right? We have to start somewhere, right?
Yeah, exactly.
It's
And
like
I
and
think,
see what comes out of it, and
and coming
then,
from physics, we try to create things with minimum assumptions.
yeah, mm,
So, like all these assumptions about random connectivity or distributions.
yeah. Mm.
and that's something AI doesn't make. So
No.
fair enough.
So yeah, exactly. So, but then if you talk about then, well, encoding, decoding, and then because how does I mean, there's also this way of I think often when you talk about modeling in neuroscience, one way to divide it up into different modeling types is sort of this like mechanistic modeling, which is sort of like the physics type forward modeling. And then you have maybe like descriptive or statistical modeling,
Yeah,
which is sort of about maybe getting representations for the data. And then you also have, and I guess both of these, that's also some kind of, I guess forward modeling is a way to model the encoding. And then you also have then, of course, decoding, but then you also have but then you also have this normative modeling, which I guess is sort of this when you try to, for example, well, there's sort of like based on this Shannon, this information, what's called information theory
yeah,
of Shannon, that you try to quantify the information contained in, for example, spike trains, right?
yeah. So
How does that fit into the overall picture
I mean, normative hasn't really come a lot in neuroscience, I would say.
here?
But a lot of work is on mostly this generative type,
Mm.
the
Forward
probabilistic
modeling,
thing.
yeah,
Yeah,
forward
forward
modeling.
modeling. Uh, I mean, forward modeling and inverse modeling, I think they go hand in hand because on one hand, you talk about these generative models,
Mm,
probabilistic models of spike trains given some inputs,
mm,
but they are also based on inference. So I think it's both.
yeah. I guess one
Um,
example of normative modeling is, I mean, there was like this work by, I guess, with Joe Attic, in where he sort of looked at, and also where they sort of looked at what should a receptive field
yeah.
look like in order to convey most information about the outside world. And I guess also there was like this sparse coding from what's it called this
Yeah, that's
1996
the Olshausen
also,
field.
yeah, field exactly. Also was some kind of asking the question, what should the code be in order to and anyway and to be sort of convey most of information about the outside world, but still not find too many spikes. So there was like with that constraint, they ended up with something that looked like this simple cell receptive
Yeah,
field.
exactly. Also very sparse responses.
Yeah, yeah.
Mhm.
So
Yeah,
that's
so there are examples. There are
mm,
definitely examples
mm,
of this kind of normative approaches, but
yeah,
I think most of the times we are talking about these generative models or more mechanistic models.
yeah, yeah,
And I try to put myself more on the mechanistic side than when I
mm,
focus on the aspect of neural code, depending
yeah,
on the neural hardware.
yeah.
Because
I think it's
eventually,
also. I mean, I also work on the forward modeling side, and it's also because that's with my physics training. I used to do like well, I mean, that's sort of what we've been trained in,
yeah,
like
exactly.
the forward modeling. Right? So the decoding has much more that sort of like other trainings, like maybe signal analysis, and that's like more other applied math trainings, which are maybe more suitable
Mhm.
for those
But
people
also,
working
I
on
think
that.
it's for neuroscience. I think at least to me, it always made sense that eventually I want to explain brain function in terms of the particular hardware the brain has because
That's
it
true.
must impose constraints on what is possible.
Absolutely. Yeah. Yeah. So, but correlation codes, because I mean, one you mentioned that examples of possible correlation codes, something that goes beyond just this independent spike code, could be like different. For example, if there's information in the detail into spike intervals that goes beyond what's in the rate. But then there's also this thing called phase coding. So, what is phase coding?
I want to go. Can I step a little bit before
Absolutely.
that?
Mm,
And so I think when I said that, there are these two extreme cases, like
mm,
there is rate code, there is this pulse packet.
mm.
In between, I think we can put one more possibility for a code, which is called sequences.
Okay.
So recently, when it has become possible to record hundreds of neurons simultaneously, what people find is that in many tasks, neurons organize their spiking.
Mm,
They are not correlated in the pulse packet sense that all of them spike together, but they are not independent in the rate code sense. They're in between in the sense that they form nice sequential activation patterns.
okay.
And that is becoming very interesting observation now. So because it turns out that there are small changes in the neural circuit architecture you can make, and then these kind of sequences, they become the most uh, viable response of a network. Also, even down to the level of individual dendrites, neurons can detect these sequential changes.
Mhm.
So, given that, as the technology is advancing, we are recording more and more neurons. I think this sequence idea is becoming getting a lot of traction now.
Ah, interesting.
So, in sequence, what we look at is that how the spike of one neuron is with respect to spike of another neuron. So, if we think of cross correlation, then we are talking about correlating two neurons and looking at not the zero lag correlation, but a non-zero lag correlation.
Mhm.
That instead of spiking like this, they spike. Now, here the neurons act as a reference to each other,
Mhm.
but it is also possible to think of another reference, and that's where the phase coding comes in.
Uh-huh.
Because it turns out that
Ah,
when you
very
look at,
elegant transition to face coding here.
thank you.
Okay, face coding. Yeah.
So, I mean, as you very well know, like at the population level, we have a lot of rhythmic activity in the brain. In
Mm.
some brain areas, it's very prominent.
Yeah.
In other brain areas, you have to put
Because we see
appropriate,
it in this local field potential
yeah,
typically.
in the local field potential.
Well, the slow frequency part of the
And if
signal
you
you
do the
mentioned.
spectrum, then you see some peaks there. Like sometimes they are around 30, 40 hertz called gamma. Sometimes it's lower, like theta around 8 to 10 hertz and so on.
Mm.
And an interesting observation is that spikes of individual neurons somehow they get aligned to specific phase of this population oscillation.
Mm.
So in that sense, you could say that the reference for the spike time of a neuron comes from the average population activity.
Mm.
So,
And that's
and
that's typically in the like hippocampus is maybe the prime example, and also maybe the entorhinal cortex.
yeah, so
Yeah.
these are two best examples. And I think here you don't even have to do any processing. On
No.
individual trials, you see that spikes are following certain phase. And in fact, in hippocampus,
Mm.
they precess, right? That's the phase precession. So you look at the theta wave.
Mm.
As the neurons start to spike in its place field, the tuning curve, if you wish. So spike goes earlier and earlier in the phase of the theta.
Mm.
And
Mm.
it's a very, very robust observation.
Mm.
And then people argued that, okay, if this is the case, then why rely exclusively on spike count? Why not take the time in?
Mm.
And then you can do better decoding. You can be more precise where exactly you are.
But one way to look at that is that, I mean, of course, we have our group. We have worked a lot on this local field potentials and how they, and of course, the local field potentials they typically stem from, at least in the cortex, are like synaptic inputs from other neurons, right? So the LFP you measure can be seen as sort of like some kind of measure of the average firing rate of presynaptic neurons sometimes some like a little bit in the past.
Mhm.
So then sort of it's more about, so the LFP actually it's a way to do, you get more spikes, information about more spikes, right?
Mhm.
So then it's not really clear that it's not really, it's
Yeah,
just we are sort of like, it's like the LFP is a proxy for a bunch of other spikes, and then the interpretation becomes, then it's more like some kind of firing rate, something that
exactly.
and so then it's like, then it's not a, then
Then it's
it's
not a reference, if you wish, right?
no, exactly. So then it's more like it's like another way to phrase the observation pool, which
Yeah.
just that takes away some of the mystery, that is not
So I
completely
guess the
different.
key idea here is that whether local field potential is locally generated
Mm. Yeah,
or it is imposed by input coming from outside.
yeah,
So in hippocampus, I think people believe that at least the theta rhythms, they are not locally generated, but are received from subiculum, for no septum, medial septum.
yeah. I know that's been in our my friends Marianne Fien in Oslo. They
Yeah,
have in that group, they have studied that, and I think that was the, I should have remember this now, but I think that was also the case for the input to the entorhinal cortex.
right.
I think, yeah.
So in that case, you can use it as a reference, but like other oscillations, like gamma, I think in most cases is locally generated.
Yeah.
And in that case, when you say, oh, my neurons are firing according to gamma phase, that's problematic, I think.
Yeah, but I mean, it means that it means that the gamma oscillations, then the gamma part of the LFP is actually reflecting the neurons, other neurons that you're not able to pick up with the same
Exactly.
electrode. So you sort of get it. So there's some kind of
It's more vague,
population
I would
code.
say.
Yeah, it's a like yeah, maybe yeah. Cool. So now of course now I'm going to change. It's like another way to talk about coding, and like you have like a term like efficient coding.
Ah,
Then you sort of, I guess that's more like a meta perspective. You think about what should be, yeah, how would be a smart way for the brain to organize the signal transmission and the coding so that it actually serves its purpose. So I guess that doesn't really say anything about whether it's rate or correlation or whatever, right? It's more like
no,
a
it doesn't. It's more a normative approach again, that
yeah.
you bring it from outside and say that, you have to represent this external world,
Mhm.
and to do it efficiently, you need certain kind of activity statistics.
Yeah, exactly. So that's like but and you had this there was just this recent paper from your group about this where you took this, I guess, this efficient coding perspective, didn't you, to look
Well,
at
not
sort of
quite. We were
okay,
basically
tell about the
asking.
work. Mm.
So we were asking. I think we were mainly intrigued by this fact that if you look at the tuning curve, so staying within the spike count code,
Mhm.
um, different brain regions seem to have different shapes of tuning curves. Like visual cortex, you have this classical Hubble visual type single peak tuning curves.
Single hump. That's like one particular
Yeah, like a
orientation, which is up.
Gaussian.
Yeah.
Yeah.
Mhm.
We were talking about hippocampus. They're also at single peak,
That's a
most cases,
play cells,
place
right?
cells.
Mhm.
And this is true for other brain areas as well. But entorhinal cortex, for some strange reason, it has periodic tuning curves, grid cells.
Yeah.
Uh, if you go to olfactory system, there we don't necessarily know how to arrange the responses on a continuous axis. So there you would say that the tuning curves are pretty much aperiodic but multi-peak.
Mhm. Mhm.
So we were asking this question like, why this diversity? What does it mean? And there is old work of Shannon in a completely different field, which has been brought back into neuroscience. Um, and the group of Lafayette, they very nicely showed that when you have tuning curves, which are like periodic, for instance, in the entorhinal cortex, grid cells, they are very, very accurate as
Mhm.
opposed to single peak tuning curves. And the notion actually goes back to,
So
to
they're very
um,
accurate in terms of the decoding possibilities.
decoding possibilities.
Yeah. So
And
there's more information. Yeah.
by analogy, also encoding that means. So you can
Yeah.
be very precise, let's say to millimeter precision, as
Mhm.
opposed to single peak,
Yeah.
which will give you a precision of a centimeter who
Yeah.
knows.
I see.
I mean, when we see the geometry of it, it makes so much sense that this
Yeah.
is how it should
Yeah.
be.
So then, of course, the question is why doesn't all tuning curves look like this?
Exactly. So
Yeah.
this is what we were worried about. And
Uh,
it turns out that when you start thinking about these tuning curves in this geometrical sense, and what people talk about, encoding or information or like precision of the tuning curves, they look at something called Fisher information.
mhm.
Um,
Can you give like a quick
it's the
pedagogical tour?
derivative of, you can say that it's the derivative of the maximum likelihood given the tuning curves.
Okay, and then
But
okay,
in simple terms, you could say that this is the derivative of the tuning curve shape.
okay,
Um,
that we like. Yeah.
so now the issue is, of course, if this is the case, then everywhere it should be like this. But when we look at the geometry of these tuning curves, so when you have multi-peak tuning curves, they kind of make some spirals in the space. Like if I take two neurons and plot them against each other, they may form a spiral kind of.
So what? How did they form a spiral? Like,
If I
in what
have,
sense do they form a
they
spiral?
will have different phases, and if you have,
Ah,
in any case, what
okay.
happens is this: that there are two kind of errors that can occur with tuning curves.
Mhm.
So no, I think I need to think of like that. Anyway, something we call as local errors, small errors, and something catastrophic, with really big errors. So, like I show you a bar 90 degrees, and you can say, yeah, maybe it's 85 degrees. That you will say is a small error.
I can live with
But if
that.
I show you this and you say,
Yeah, it's
that's
like
bad,
vertical.
that's
Yeah, like a
catastrophic.
horizontal instead of vertical. Yeah.
Yeah. Turns out that these grid cell like tuning curves, they are very susceptible to these large errors.
Uh huh.
So they reduce small errors,
Okay.
but they increase the probability of large errors.
Ah.
Now, when you do the statistics, because small errors are more likely, the whole
Uh-huh.
thing is biased towards small errors. And then you say, oh, grid cells are very, very precise.
Ah. Hmm.
So we focused on this idea of large errors. And the
Okay.
argument we made was the following that, um, as a monkey, of course, you want to minimize this error, but you don't want to miss a branch.
Oh.
Or, I mean, a catastrophic error is really catastrophic
Yeah.
sometimes.
Hmm.
Um,
I see.
I feel like bringing in a cricket analogy now.
Yeah. Yeah. Exactly. You're Indian, so so it's like cricket is your national sport.
So, like one sport where we really worry about orientation is cricket.
Okay.
The ball position matters a lot, whether the ball will move like this or like
Yeah. You
this.
have to. We are on radio now, or we are podcast, so you have to show. So yeah. So you can move whether you're
I
vertically
can
or so.
show you the ball,
Yeah.
which is
Okay.
always handy. So you
Okay.
need to see
Now
this
we see
orientation, if
cricket
it is
ball
like
here.
this or
Yep.
this.
Mhm.
And if you miss it, you miss
Okay.
it. And you
Okay.
have to do it very, very fast because the ball is moving very, very fast.
Okay.
So we asked this question like, how much time do you need to collect enough evidence so that you can avoid these catastrophic errors, the
Okay.
large errors? And there, when you compare the periodic and the single peak tuning curves, visual system versus entorhinal cortex, it turns out that visual neurons are very good at minimizing these large errors. They can make a small error,
Mhm.
but the probability of large error drops very rapidly.
Mhm. Yeah.
Entorhinal cortex neurons, by contrast, they need a lot of time
Yeah.
to avoid catastrophic errors.
I see.
So, in a slogan-like sense, I could say that visual neurons maybe are sloppy, but they are very quick,
Ah. Yeah.
as opposed to entorhinal cortex neurons. They are
What do you
meticulous,
mean?
but
It's
they're slow.
slow. They're slowly, but what is it now? They are.
Slow meaning that you need certain time
Yeah,
to
they're
collect
slow.
enough spikes.
Yeah, but you would say that they but slow but quick. You said no. No. What do you
No,
mean?
I said sloppy.
Sloppy.
They are
Sloppy.
sloppy but quick,
Okay.
and the
Okay.
entorhinal would be then meticulous but slow.
Okay. And also this chance, I was thinking of like AI, right? Some worries about this deep learning that they can do these catastrophic errors that no humans would do. So that's like the grid
Okay, yeah,
cells
that's
also
another thing, right? There are these adversarial examples.
example of what I mean that you
Yeah,
just do these really silly things that no humans would
exactly.
do.
So
They
that's
can,
really a catastrophic error.
yeah, exactly. So that's so the grid cells, they are like the AI. Okay. Anyway,
But
no,
only in short time. Like if you
yeah,
put pressure
I
on
see.
the animal to quickly decode, let's say with grid cells,
Yeah,
they are gonna make large errors.
yeah,
But if given the time, they will
yeah.
perform better than the.
Excellent. Cool.
And this gives a very nice perspective now that why. So one constraint is the amount of information you can decode or
Hmm.
encode using
Yeah.
tuning curves.
Mm.
Other constraint is how much time you need to recover that
Yeah.
information,
Mm.
avoiding these large errors because they are really survival
Because I
threats.
guess, as you mentioned, the olfaction, which has these multiple tuning curves, that's a very slow sense, right? So then you have enough time to come to a sensible conclusion.
Yeah, I think so. That's what I
Yeah.
would. But I don't want to say a final word on it yet
No,
because I think part of the reason why it may be slow is also because the signal transduction could be slow.
that's true. It's like diffusion of
Right.
molecules and
So
up to
that
the
could
one.
also be a reason. And I
Yeah.
know that our friend Upi Bhala, he
Yeah.
actually showed that actually speed could be quite good for olfaction as well.
I see. I see. Wow, that's cool. So this is, I think, the first author is Leninger. He was a
Yeah.
student with you, right? And
So
that's
Momitz
in.
is a PhD student at the Information Science Division.
And
So
he.
he and Mikkel Skoglund, they are the information theory experts.
Excellent. So
And
that's
Pavel and I, we are the neuroscientists in this
exactly.
team.
I would
So it was
love
a
to have.
nice combination of the two
Yeah.
expertise coming together.
Yeah.
And but yeah, Movitz led this work. He posed this problem in terms of time,
The
which I
Moritz
think,
is Leninger. Yeah.
yeah. So
Mm,
he basically asked this question: Hey, why do we ignore the time aspect of it?
mm, mm.
What
Yeah.
about that? Time is important for survival in the natural environment. So
Mm,
he posed this problem and then he solves it.
mm,
And he finds two important results. One is this thing about single peak versus multi peak.
mhm,
The other thing he finds out is that this problem becomes even worse when you have high dimensional stimuli.
mm,
So I was only showing, the seam of the cricket ball, for instance.
mm.
but you also want to know which side is shining,
Okay.
right?
It's not easy to be a batsman in cricket.
No, it's not.
That's it. I guess you have 1 billion Indians were training to be the best one to come on the
But one day
national
we
team.
will talk about why it is difficult to be a bowler also.
Yeah, well, that's that's hard too.
No,
The
but I
bowler, yeah,
think
yeah.
why I'm referring to sports, I think for human experience, I think sports provide this a nice experimental condition where you have to act very, very fast.
Mm,
And
mm,
you have to integrate many different information. I guess
yeah,
the same is true for like a ski jumper or somebody who is doing downhill skiing.
yeah, mm,
Perhaps even more difficult given the
yeah.
contours of the
That's
snow
true.
and
That's yeah. That's true. On the other hand, you have like this in cricket and in baseball, you have these adversarial attacks, right?
yeah, exactly,
You want the bowler to make it as hard
exactly.
for the batter as possible. So,
So the thing that he said was that actually this problem becomes worse
mm,
when you have high dimensional stimuli,
mm,
meaning neurons are caring for many, many dimensions. Then
mm,
it becomes much worse. You need even more time. So this then makes us think. Now, this is a very bold proposition that maybe what grid cells are doing is very low dimensional representation of something high dimensional.
mm, mm.
And maybe they are actually not representing space, perhaps some other cognitive aspect of the navigation. And actual place representation is actually taken care of by the place cells.
Mm,
And there
mm.
is some kind of dependence. I guess you know it better than I do, like whether place cells give rise to grid cells or vice versa.
Yeah, I'm well. Yeah, actually, I did some work on how you with grid cells how we can make play cells. But that's a long time ago. So I think there's sort of like I'm not quite sure
Okay,
if the verdict is out on that, or I haven't followed it so closely. But there are
so
certainly
that's
connections both ways. So
so that's another aspect. And it turned out when we wanted to find some experimental evidence of this argument about high dimensions, turns out people don't do these experiments.
mm,
So
mm,
most of the experiments for visual system, for anywhere, like you give very simplistic stimuli to the animals to make it
mm,
easy for them to work
mm,
right in the experimental setting.
mm,
But in real world setting, you have to deal with.
yeah,
So one of the strong suggestions that we make then is that people should be doing these multiple dimensional stimuli experiments. And other thing that was detrimental for this decoding and catastrophic error is background activity.
okay,
Uh, Background activity has many interesting usages, but in this case, higher the background activity, higher the chance of making these catastrophic errors. So either you need a very large population or low background activity.
yeah,
What
mm,
nicely fits is that entorhinal cortex is amazingly silent place,
ah,
as opposed to the visual cortex, which has
yeah.
fairly high baseline or ongoing
Of
activity.
course, in triangle cortex we have the grid cells, and
Yeah.
mm,
So that I think there's a lot of these different ideas they are falling in place
yeah,
and trying to give
mm,
us some framework to
mm,
now think about why there's diversity of tuning curves across the brain areas.
mm. But I mean, here is sort of like I guess this was sort of like idea of efficient coding, at least in spirit, right?
Yeah.
I mean, this is sort of how can this maximal information or how to
Yeah.
deal with the environment, and then you have another, and I guess sparse coding. That's that is maybe so that's the constraint maybe that spikes or requires a lot of energy, a lot of food. So you would like some kind of that's another principle that is sort of also a little bit like a normative in a sense.
Yeah.
How does the coding? How can you make the coding so that the animal has a higher chance
Right.
to survive? So that's more like an energy constraint, maybe.
Funnily enough, sparse coding also arises when you impose some constraint on synapses.
Okay, what kind of constraints and
So synapses, we typically say that there's a synapse which has some weight, but
mm,
as you know that they are functions, so they are some transients which
mm.
follow, let's say, some exponential family.
Yeah, exactly. So if there's an action potential
But
coming in on the presynaptic side, there's a little bit of a current
but
that lasts for a few
they are not
tenths of a millisecond.
fixed. They change, not in long term, in short term. So something called short term facilitation or depression happens.
Mm,
So
mm,
if we use a neuron, a synapse, again and again, the amplitude of the spike response becomes smaller or smaller, or bigger or bigger. This is if it becomes bigger, we call it facilitation. If it goes smaller, we call it depression. So it turns out when your synapses are depressing, at that point you want many neurons to respond but very few spikes. Otherwise, you will lose them because synapses will depress.
Mm,
If you have facilitatory synapses, you want your spikes to be on few neurons at a high rate so you can exploit the facilitation.
mm,
So it turns out there is this extra constraint comes from synapses,
mm.
which tell you whether you should be sparse or you should be dense,
Yeah.
whether
So
you
there
should
we're
be low
back to
rate
this thing
or high rate.
of not having a brain perspective on decoding
Yeah,
instead of a human
more a
perspective.
forward perspective, right? If
Yeah.
you wish.
Yeah. Mm,
So this, I think, is a nice way to convert to synthesize this, and hopefully, like in Perkel's book, we have a lot of possible candidate codes.
yeah.
And I think normative is one way to put boundary conditions on
Mm,
them, and the neural hardware is another way to put boundaries
mm,
on them. And hopefully, in the end, we will end up with few
yeah.
examples.
But then another one is like predictive coding, and that's maybe not normative in it. That's more like an idea about how the signal processing, well, I guess interaction between the top down, like, and bottom up, and like how like the sensory input versus sort of like what is already stored in the brain, and how that is. What how would you talk about predictive coding?
So yeah, I'm a little bit uncertain about this topic.
Mm,
Um, I mean, yes, we do predictions,
mm,
right? We do expect anticipate things and check against the evidence. So it looks like a very nice way to work in this environment, and it massively speeds up the things,
mm.
right? If
Because,
you really have to react, you'll be very slow.
because I mean the idea with predictive coding is that like this when you sort of have like sensor input, then a little bit like at the quite early up in the brain, you sort of meet the expectations, and
Yeah,
then there's some processing done. So what is really transformed to higher stages like the deviations from the expectations is
yeah.
that's like or the errors in some sense in your
So at the
prediction.
behavior level, this is the idea, right? Like if you are on downhill ski, you
Mm,
anticipate like snow will be soft or hard, or there will be a curve coming or something like that, right? That's one way. But and that works very well in motor system. There is sufficient evidence for that. But in this sense, what they do is that they are not really predicting the future; they're predicting the present.
mm,
And
mm.
this is where I somehow get confused.
Okay. Yeah.
That what are they predicting? There's no prediction; it's just the current estimation.
Mm,
So
mm.
I don't want to say too much on this, but I'm a
Uh,
bit confused about predictive coding.
maybe it's the word you don't like that.
Yeah, maybe it's that. Yeah,
Yeah, exactly. So it's like
I mean, in control system sense, right? When we have a feedback control system,
mm,
there we never talk about predictive coding or predictive control in that sense.
mhm,
But here it seems we do, and the argument is that top areas like PFC etc. they make predictions,
mm,
but I mean the space of predictions is huge.
mm,
How do you know what to predict, right?
mm,
So
mm.
yeah, I mean, I think let's say normatively or like in a generative model sense, not normatively, generative sense, it makes sense.
Yeah.
From the bottom up, like really hardware side, I think it's very tricky to imagine this.
Okay. So we're approaching the end of this podcast, and I guess one thing that I mean, we see. I mean, you have like argued that maybe you have some rate codes, and I guess, but still, if you decide that it's a rate code, you have this question about tuning curves, like this paper that we mentioned with the difference between single
Yeah,
peak tuning curves and multi peak tuning curves, and but then also you mentioned this that there might be that the code might really vary with where you are in the system. I guess there's both in the I guess the auditory processing in the barn owl, and also I think in the spiking of this. Well, I think there was like the group of Rasmus Peterson, I think in Manchester, who found there was like a microsecond precision in the timing of spikes in this. I guess it's like the what's called the trigeminal
yeah,
primary afferent in rats, but it's sort of like the analogous thing to the retina, I guess, but it's not in the whisker system. So they have some places where you have this remarkable temporal precision, which then probably also then it's like well, I don't know if then it's really like well, time seems to be of the essence. I guess it's like temporal code of some kind, right? So I yeah.
I
Mm,
mean, it does seem like that in the periphery you can have very precise time codes.
mm,
But whatever we have learned about the neocortex, at least,
mm,
using models of the brain, different levels like microcircuit or network or mesoscale, turns out that the ongoing activity or ongoing noise and correlation, they impose a severe challenge for
mm,
time-based codes.
mm. So maybe like a preliminary. Of course, we are scientists. Everything we like science change when the new science ideas change when there's better science coming around. But the preliminary idea is that maybe for the cortex, it's mainly rate codes and maybe in in like out in the periphery sensory system, like especially out far in the
Yeah.
periphery, then it's more maybe like time codes and other kinds of.
I think for cortex, other candidate which is more likely, and as I was saying that small tweaks in the architecture of the networks that we have been studying could enable them to generate sequences.
Yeah,
I think
yeah.
sequence perhaps is a more likely scenario.
But where does
No,
that fall on the axis between a rate code and
it's
a
in between.
temp
I
in
think
between?
now again the issue is about precision, right?
Mm,
How
yeah,
much flexibility is there? Meaning
yeah,
a given population, how many
exactly.
sequences can it generate?
Mm,
There is all kind of interesting questions there, but initially, like I would say, like 10 years ago, we would have said that hey, sequences are very tricky to get in
mm,
recurrent networks. You
mhm,
need specific learning and wiring and so on.
mhm,
Like we embedded feed forward chains manually pretty much 20 years ago, 15 years ago.
mhm,
So that is, I think, gone now. So you can tweak some things in the neural architecture and you can exploit the asymmetry of the neurons. Neurons are not symmetric objects. They
mhm,
will have preferential connections in some directions, and that could make it much easier for networks to generate sequences.
mhm,
Similarly, all these attractor ideas, combine them with short-term dynamics of synapses or neurons, you can get moving activity bumps.
mhm,
I think those could form a nice intermediary between precise time codes of Moshe
mhm,
or absolute rate codes of Adrian.
mhm. I see.
Uh,
Huh. So the new things happening on the Adrian Ovilus axis.
I think so.
That's cool. So, I mean, now one thing that we see now, it's, I mean, they talk about the Moore's law for spike recordings, right?
Yeah.
That it's, I think I don't know now. You can easily record. Well, I guess that's. I mean, with the electric recordings, you can sort of easily now record much more than 1,000 neurons. At least if you have many like this neuropixel probe that they make. Which has sort of like where you can maybe record from like 100 for on each probe, but then you can have many of those. And then with this calcium imaging, you can also record. Of course, then you don't get the time result each spike, but then at least you can get more and more. Well, you get a really large number of neurons. So I guess it's. Do you think that is going to be important for? So how important do you think that's going to be?
Hmm. The problem is this now, and maybe I'm a little bit too opinionated about it
Well, that's
without having worked with this
yeah,
data.
towards the end of the podcast, you're a little bit can be a little bit freer in our be a little bit more opinionated.
So what has happened is that I think the focus has moved from this actual question about neural code, etc. People are more and more interested in decoding. They
Mm,
don't care how exactly.
mm,
So
yeah,
pretending to be an ideal observer, so fitting regression, just doing a regression on
mm,
stimulus variables and the neural activity, record activity in 1,000 brain areas,
yeah,
record 1,000 variables of the behavior, do a regression.
yeah. And then sort of like to see sort of define neural representations
Exactly.
for all kinds of things. Mm,
And I don't know how helpful that is, because I mean I'm never surprised when somebody says, hey, this particular variable of the stimulus is represented. Yeah, of course, I mean behavior doesn't fall from sky.
uh,
It
uh,
comes from the neurons somehow.
yeah, mm,
So it is a nice way to discover some unexpected representations, like, oh, this area is also involved in this task.
mm.
So, like
Mm.
for instance, we are in this phase of neuroscience where every brain area process everything.
Mm. Yeah.
Uh, So this is actually a nice phase in the sense that you get some unexpected results that, oh, this brain area is also contributing to that function, and that could facilitate building of like nice algorithms of processing.
Mm.
But the focus has moved from extracting this so-called neural code.
Ah.
Uh, there are good reasons for it. Rate code is trivial. I mean, you don't care of how many neurons you have, but when you go into the time territory, beyond 10 neurons, you have so many statistical tests to do
Mm.
that there will never be enough trials or,
Mm.
and I think that's a big problem in terms of this detecting time code.
Mm. What about like I mean, there's like a lot of attention also on these neural manifolds,
Hmm.
which is a way to, of course, then you sort of look at the firing rates
Yeah,
of different neurons as a function of time in this high dimensional
yeah.
vector space. Mm.
So that's actually a nice development. It's a better way to go beyond, better way to look at the data and go beyond individual tuning curves, because
Yeah.
essentially manifolds are also a form of tuning curves.
Mm.
But now instead of seeing individual tuning curves, calling them single peak, many peak, you just
Ah.
call them a manifold of some
Ah.
sort.
Huh.
And they have very nice properties, good in, yeah. That's
Mm.
actually nice
Mm.
development. However, it also ignores the time aspect.
Well, I mean, I guess you could sort of, I mean, this you could move around
Yeah,
in this high dimensional vector space as a
you
function of
can,
time.
but to
That's
create something which is continuous manifold,
that's
you
smooth.
need
Yeah.
sufficient time resolution, like
Okay.
sufficient number of spikes.
I see. So in practice, it's not so. You need exactly. So you need a firing rate for each neuron, and then
Exactly.
often you need some kind of
And typically, if you will
spike
read these
counting.
papers, they will talk about bins of like 50 millisecond
Okay.
minimum,
Yeah.
maybe even more, like hundreds and so on.
So
So in
more like
that sense, it's a bit time. In terms of time resolution, it's very coarse.
so it's
Now, this idea is, I I don't know if Peter Latham remembers it, but
mm,
once Peter Latham said that all you can do about the brain is decode it,
mm,
and I think if that's the goal, this is
mm.
actually a nice development.
Huh. Hmm. Okay. So now I know this. You had this nice paper out. This was a Lenninger paper, which is out in eLife, and I'll sort of put the link out on the podcast webpage. So is this? So what's your? I mean, you work on many things. I know in your group in it at KTH, the Royal. What's the Royal Institute of Technology and Science?
Only technology, we don't do science.
Okay. Okay, I see. So this is in Stockholm. So then you
Although,
have
although the institute logo says Kunst och Vetenskap,
yeah, no, but
we do
exactly.
art also.
Yeah, no, but the Swedes have just the Swedes have always sort of like back in the days when they were really like almost like in, had some imperialistic ambitions. They wherever they went, they both brought weapons and knowledge. Right? They
Okay.
started universities like in the and so I think in like Estonia, for example, as I came across a university that was I think it was in Tartu or something,
Uh-huh.
started
Okay.
by the Swedes. So they had like this. Anyway, so but that's good. So, but what's your plan now? For do you want to work on coding issues?
Yeah. So, I mean, this paper. So we are extending this idea now to ask other questions. For instance, everything here was done assuming uncorrelated spikes, and we touched upon the issue of correlation. So
Mm,
we are curious, like, I mean, it's not feasible to be so slow using tuning curves. I mean,
mm,
that's not good.
mm,
So we are hoping that correlation somehow help in this.
mm,
So that's one direction we are exploring, that how could correlations be used to accelerate the decoding and avoid catastrophic errors. But I mean, I go back to my old interest in transmission. So I'm also interested in like transmission of information now. So far we only talk about pulse packets, which is 0 or 1 pretty much. There's no information or
mm,
full information.
mm.
So something graded, and I guess this is a very nice paradigm that you have some representation in one layer, then the next, and the next.
Okay. So
So how
this is
does this?
why you talked about sequences. That's maybe one aspect you're interested in then.
That's also possible. So, but I
Yeah.
think in visual system, there's not so much of sequential activity observed.
Yeah.
But for visual system, really, like the other question is with regard to this. everything is feed forward here. We never made any assumption about like how neurons interact with each other. So that's
Mm.
another thing that has to be studied.
Yeah.
There's a very nice question that one can pose here is that why neurons with similar tuning are clustered. So this is an interesting feature of primate and the cat brains, that visual brains, that neurons with similar tuning are put together in pinwheel pattern.
Mhm.
I don't know what's the advantage or disadvantage of that,
Mm.
but it must play some role because at least these animals have much better vision than rodents.
Yeah. Mm.
So there are some nice questions that we hope that we can address using this particular framework of the geometry of the tuning curves as well as this notion of catastrophic errors and
Yeah.
time.
Cool. Well, looking forward to see the development of that. So, thanks a lot for coming to the podcast, Arvind.
Gaute, it's always a pleasure talking.
Yeah.
Thank you so much.